Let's talk about a huge bottleneck in life science research: prepping cell samples for analysis. You know the drill, right? Traditional methods can be harsh, slow, and just so inconsistent.
Honestly, all that manual prep can really tank the quality of your final results.
Yeah, these methods are a ton of work. We're talking multiple, complicated steps that take hours. But what if there was a better way? Something faster, gentler, and way more consistent.
It actually levitates cells to separate them. No labels, no harsh forces. Super cool, right?
It takes this super complex task and makes it simple, with a totally automated workflow. Think about this. Your hands-on time goes from hours down to just 5 minutes. Wow. And here's the real power move. It can process a whole 96-well plate at once. And this is huge. It's label-free. That means it's incredibly gentle on your delicate cells. Because it's so gentle, you get fantastic cell viability, usually over 95%. Plus, automation means less variability, and it's amazing at clearing out all that debris. So when you put them side by side, the advantages are just obvious. It's a no-brainer.
So what's the big deal here? It's not just about saving time, it's about better research.
This means cleaner data, sharper results, and a streamlined workflow for the whole lab. Because at the end of the day, it really comes down to this: better samples, better science. It really makes you think, what could you achieve with purer, healthier cell samples?